My name is Abby and I'm a part of the Olive Honest Institute team, and we really do appreciate you guys taking the time from your busy day to learn more about nutrigenomics, the Mediterranean diet and extra virgin olive oil. Just a few housekeeping points I need to make. So after the presentation from both Flavia and Carlotta, we'll have a short question and answer session. So please feel free throughout the presentations to write any questions that you guys may have in the comment box on the bottom left-hand corner of the page. So we will endeavour to get to as many questions as possible, but of course, as time is quite limited, as we've got two presenters, we might not get to all the questions, but my team and I will make sure that we see all the questions, we take note of them, and we'll make sure that they're answered, and we can send them the questions and answers to you guys in a post-webinar email. There's also going to be a short survey that I'll pop up at the end of the presentation. We'd just appreciate if you guys could take the time to complete it. It's pretty short, only 5 questions, just helps us improve and continue education opportunities. We also have some exciting news to share with you guys at the end, so stick around for that. So I now want to welcome our first presenter for the day, Dr Flavia Fayet-Moore. She's a registered nutritionist, accredited practicing dietitian, board certified lifestyle medicine professional and honorary associate of University of Sydney, and is also the CEO of Nutrition Research Australia. So welcome, Flavia. Thank you so much for your time today. I'll now pass over to you.
Thank you, Abby. So thank you, everyone, for taking the time out of your busy schedules to come to this webinar. I know that you're probably having lunch while you're watching this. Now, I'm extremely passionate about nutrigenomics. It's an area that I've been involved with for nearly 20 years. I did the very first course of nutrigenomics that was offered by the University of Toronto in Canada. And since then, I incorporated nutrigenomics as part of my PhD. Have been using it in clinical practice for over 7 years. And I'm now working with Nutrigenomics as a consultant as well. So basically what I want to do is give you everything that you need to know from my 20 years experience about nutrigenomics in 25 minutes. So let's go. So to start off with my disclosure, again, as I mentioned, I'm on the advisory panel and consultant to Nutrigenomics, the company. And I'm also on the Olive Honest Institute advisory panel. So I'm basically the lifestyle medicine, nutrition, dietetics expert on the panel. And obviously you're aware of the Olive Honest Institute, the repository for everything on olive and wellness, because you're sponsoring this webinar today. So to get started, just to give you an overview of what I'm going to be talking about, I'm going to define nutrigenomics or nutritional genomics, talk about some of the current applications of it, give you an overview of the science with some examples of where it's at, then talk about the impact of behavior change of that science in nutritional genomics, talk about some tech offerings of what's available out there, and then tell you what next, where are we going from here. Now, the concept of nutrigenomics, or the concept that one man's food is another man's poison, has been known for a very long time. It's not new. This statement, one man's food is another man's poison, was known since the 1st century BC. So for a very long time, we know that people just respond differently to different foods. Some foods or supplements are good for some people, and for others, not so good. So just to give you a recap, as I mentioned, I did the very first course nearly 20 years ago, and at the same time as I was doing that course, the human genome, or the blueprint of the mapping, the sum of all our DNA, was finalized. So this was in April 2003. Basically, that was finalized. So it's quite recent and new, the science of nutrigenomics. But so is the science of nutrition when you think of it, because the first vitamins and minerals were only discovered in the late 1800s, early 1900s. So nutrition as a science and nutrigenomics as a science are quite new. But technology is becoming better and quicker, which allows us to get information and build that knowledge a lot faster. So one of the breakthroughs of the year was the fact that we have genetic variation in our human genome. So even though everyone has the same blueprint, we have different variations of that code within our DNA. And that's kind of one of the bases of why we respond differently to different diets and nutrients. So let's define nutritional genomics. So imagine nutritional genomics as a coin, and there are 2 sides to the coin. Now, I loosely use the term nutrigenomics to talk about nutritional genomics, because it's just quicker and easier to say. But nutritional genomics is the coin. And then on one side of the coin, you have the way that diet impacts the way your gene works. So that's the nutrigenomics as a definition. And on the other side, you have your genes that influence how you respond to diet, and that's nutrigenetics. So basically, you can have diet or nutrients or supplements that alter your genes. So for example, there could be components in broccoli that make your enzyme in your liver work faster. Or there's the genes that influence how you respond to diet. So for example, there might be a gene that codes for how quickly you might excrete a certain nutrient. So you might not absorb it very, very well because a lot of it will be excreted. So that's the overview, simple explanation of the science. Now, the genes can affect lots of different ways of how we respond to diet. It can code for how you smell food. And, you know, as everyone knows, when you have a cold and you can't really smell food, it doesn't make it very appealing. So if your genes influence how you smell food, it will influence how much of it you eat it, if you like it. There are different genes that code for taste. So some people have a code for the bitter taste receptor that is expressed more. And let's say if your mom or your dad is trying to introduce foods to you, broccoli and kale and spinach, and you taste that bitterness very easily, it's going to be a lot harder for you to like those foods than someone who doesn't have those genes being expressed. So you can see that genetics can play a role not only on how you metabolize and excrete food and nutrients, but also can influence your food preferences and can even influence your food preferences from when you're first starting your first foods. So again, appetite, metabolism, it can alter how you absorb a particular nutrient, how you digest it, and even how you excrete it. So the basis of personalized nutrition is really to discover, which is the basis of nutrigenomics, is to discover your predisposition to diet-related diseases and food preferences, perhaps. Then you consult with a dietitian or a nutritionist or other healthcare professional that's an expert who will help you interpret your results using all that other information, you know, usually phenotypic information that we collect, including as well as lifestyle, so exercise, sleep patterns, smoking, and so on. And you merge those results, the genetic results, with your lifestyle preferences, and then recommend a specific dietary plan or the specific foods to help treat and prevent chronic diseases. Now, one thing that nutrigenomics is not, it's not a predictor of risk. So Angelina Jolie made it quite popular when she came out and said she got tested for the BRCA gene. Breast cancer runs in her family. She wanted to know if she had that risk predictor gene. And she did, so she chose to remove her ovaries and her breast to reduce her risk. And that's basically all she can do, because it's a gene that highly predicts an 80% chance of developing the cancer. Whereas modifier genes are actionable genes. They're all modifiable based on diet. So your risk for a disease, per se, let's say heart disease or diabetes, would be totally dependent on the interaction between your gene and what you're actually consuming. And the recommendations that you get from that are actionable. These genes are also really, really prevalent. So most of you that are watching this webinar right now are bound to have multiple genetic variants that are very common in nutrigenomics. So these are like found in 1 in 2 people, 2 in 3, 7 in 10. They're very, very common, and you're bound to have more than one. Now, what if I were to tell you that nutrigenomic testing is actually done in every baby born in every hospital in Australia and in most hospitals around the world? Would you believe me? Well, it actually is. So this is one example of nutrigenomic screening. So that's the newborn screening for phenylketonuria. So basically, the newborn has a particular gene that alters how they break down phenylalanine. They can't break it down, so they basically have to avoid it to prevent the adverse health effects. And all they have to do for the rest of their life is to avoid phenylalanine. If they don't avoid it, then they have the adverse health effects resulting potentially even in mortality. So this is a very clear example of how it's government funded. It's available to basically every baby that's born in the world, most babies born in the world. And that's something very common that we actually use from a public health perspective. And then what if I were to tell you that there are food products designed for your genes? There's actually quite a few. So there's lactose-free milk. So if you can't digest lactose, which is a genetic basis for it, then you have to avoid it so you don't get gastrointestinal symptoms. There's gluten-free for those who have celiac disease. And there's even food labeling regulations. So if you think about the PKU, certain food products that have aspartame have to have a warning that it contains phenylalanine. And that's at the food regulation level where we have policy implemented for it. Now, something to highlight is that the cost of this genetic analysis is decreasing significantly. So back when I was doing my undergrad, it cost $100 million to sequence your whole human genome. And now it's just under $1,000 to sequence your human genome. So as the cost decreases, those public health interventions and screening will be done at a population level because it will be cost effective to know those risk factors that you have and alter your diet to actually prevent disease. So the whole concept of one size fits all is basically the nutrition recommendations we have nowadays. There are general recommendations, it's not personalized, and a lot of this recommendation is based on very little or no RCT evidence because they're just inherent problems we have in nutritional research or dietetic research where we can't do RCTs. It's nearly impossible to blind people from food because everyone needs to eat from birth. So everyone has had some sort of exposure. You can read for some further reading a little bit about those papers I have up there as a reference. But basically, my point is that we use a lot of epidemiological evidence to make current nutrition recommendations. And I'll explain how nutrigenomics can help with those epidemiological evidence. Because disease takes a long time to happen, so you need to eat poorly and not exercise and not sleep and smoke for a long time because you get the disease. So it's quite hard to do an RCT that's going to last that long. It would just be extremely expensive and unrealistic to do. So in any epidemiological studies that have been done in nutrition, we all know if you correlate any particular component of our diet with any particular disease, there's bound to have mixed findings. So I want to give coffee and heart disease as an example. So there's been plenty of studies that show coffee increases the risk of cardiovascular disease, it has no effect, or it's actually protective. So the idea of nutrigenomics is, can we predict with genetics that perhaps someone with genotype A will have an increased risk, someone with genotype B will have no effect, and so on. Now, before I go into coffee and heart disease, I just want to highlight that there's lots of bioactive components in coffee. There's actually hundreds of bioactive components. Coffee is not synonymous to caffeine, although it's one of the largest sources of caffeine in the world. But there are antioxidants, there are nutrients also in coffee that are important. But caffeine by far is the most widely studied component. And we know that there's a common genetic variant called CYP1A2 that codes for the CYP1A2 enzyme that metabolizes 98% of caffeine in your liver. So there was a study done by Harvard and the University of Toronto that wanted to investigate the relationship between cardiovascular disease and coffee consumption. So they used over 2,000 cases and 2,000 matched controls. So they matched for everything like saturated fat, smoking, exercise, BMI. They collected food frequency questionnaire data on coffee. And coffee, using an FFQ is actually quite easy to do because most of us know how many cups of coffee we generally have in one day. You know if it's 1, 5, 3, and it's generally quite consistent for a lot of people. So it's a quite accurate way of measuring coffee intake. So this is the CYP1A2 enzyme that occurs in the liver. This is how caffeine is broken down. For those nerds in the room that want to see the biochemistry. So you can see there, CYP1A2 breaks down caffeine into paraxanthine, into its metabolites, and then it's excreted in the urine. Now, half of you listening today are fast caffeine metabolizers, and the other half are actually slow. So this is the inducibility of the enzyme, or how quickly it works. So if both of your parents inherit an A copy, you're going to be a fast metabolizer. If you happen to inherit a C copy from either parent, you raise low caffeine metabolizers. So that has nothing to do with how much caffeine stimulates you, That's the adenosine receptor binding affinity. There's no way to know if you're fast or slow. It's basically a genetic test. So it has nothing to do with that, how much it stimulates you, if you can sleep or not. It's basically how quickly your body can get rid of caffeine. Now, going back to that study from Harvard and University of Toronto, let's say we were to ignore genetics altogether, and you would find that basically as coffee intake increases, so from less than 1 cup to greater than 4, the risk of having a myocardial infarction or a heart attack increased significantly, only among those who had greater than 4 cups per day. Now that sounds about right. You basically, international recommendations are around no more than 3 to 4 cups of coffee per day. Now, what happens if you split this population based on their genotype? So the only difference between the two groups I'm going to show you is one group is a fast caffeine metabolizer and the other group is a slow caffeine metabolizer. So they're matched for saturated fat, for BMI, for smoking, exercise, and so on. So if caffeine is really the bad thing in coffee, you would predict that if you're a slow metabolizer, caffeine stays longer in your body and it's probably going to do more damage to your heart. And if you're a fast metabolizer, perhaps caffeine gets excreted quickly and you retain all those antioxidants and polyphenols and nutrients that may be protective. I know you're waiting for this. Ready? Here, this is what you get. It's quite clear that if you're a slow metabolizer, even at 2 cups of coffee a day, your risk of having a heart attack increases significantly. Whereas if you look at the fast metabolizers, it seems to be even a small protective effect if you have moderate consumption of coffee. So this study has been replicated multiple times from independent groups all over the world, and the CYP1A2 gene has even been implicated with hypertension and insulin sensitivity. Now, what about weight management? We know everyone's quite interested, can I blame my genes for making me fat? Well, there's actually a really quite interesting study that was published looking at the PPAR gamma. And in this study, the PPAR gamma works on differentiating adipocytes, so your fat cells. And this was a large randomized trial with Spanish overweight and obese individuals, where they used a Mediterranean diet to lose weight. Now, keep in mind that this is a Spanish population. They love their extra virgin olive oil. So the median intake of fat from MUFAs, because extra virgin olive oil is basically the only fat in their diet, was 56%, so it's quite high. What this graph is showing you is that if you inherit a G copy from either parent, you only lose weight if your MUFAs are greater than 56%. So there's that very clear interaction between your genes and your diet. So if you were to put everyone on the med diet and not look at their genetics, some people will benefit a lot, some people may not benefit as much. And if you look at the PPAR gamma gene and how it interacts with MUFAs, there's a clear relationship that you'll lose more weight, 2.2 points lower BMI, also lower fat percentage if you increase your MUFA intake and you focus on that. So it's really clear from the evidence I've provided so far that one size does not fit all. It's, we're quite individual and we respond to diet based on our genes quite differently as well. Now, the next question that I would ask is, okay, it's great to know those things, but does giving someone genetic information actually help them change their diet? So again, this is from Dr. Ahmed Al-Sahani, who founded Nutrigenomics, that has the lab in the University of Toronto that does a lot of this research. They discovered the CYP1A2 enzyme variation. They actually did the world's first randomized controlled trial providing genetic information for personalized nutrition. And the first paper they published, they found that people who received the personalized genetic information were more motivated to change their behavior, and they understood the recommendations more than people who just got the guidelines to healthy eating. Then they followed them up a year later and found that even among a healthy young population, so these were university students, those who received the genetic information, specifically for the ACE gene, angiotensin converting enzyme, which puts them at a higher risk of having high blood pressure. It means that they're salt sensitive. They reduced their sodium intake from that one consultation 1 year later. They remained with that lower intake. So it actually resulted in behavior and it resulted in adherence. Because we all know, as nutrition professionals, that advice is only as good as if someone can adhere to it. And again, another paper was published showing that it's quite effective if you give personalized genomic testing. Even direct-to-consumer has been shown to be effective at getting people to change their diet and their exercise. Now, if you want some behavior change evidence related to the med diet and genetics, this is a perfect example. This is what was a large RCT where they looked at adherence to the Mediterranean diet in the Food for Me study, which is one of the largest randomized controlled trials done in Europe, looking at nutrigenomics and personalized nutrition. So in the first group, they just gave general nutrition advice. The second group, they gave personalized advice. The third group, they gave personalized plus phenotypic advice. And in the last group, they gave the genetic. And the only group that had a significant difference in weight loss from the Mediterranean diet adherence was the group that actually received the genetic information. And that was also replicated with adherence to weight loss when you get your genotypic information in another Food for Me randomized controlled trial findings, where they looked at the FTO genotype. So people who received that information that they had that genotype lost more weight. There's also systematic literature review that's also showing that if you provide that actionable advice, that people are more likely to change their health behavior. Now, this is not going away. For those of you that think it's a fad, it's definitely not a fad. It's a hard science that is quite profitable. You know, the global nutrigenomics market is projected to grow to $17 billion, and that's in 3 years. So this is a few years old, that slide. And the personalized nutrition market is going to be $6 billion plus. So it's not going away. It's, you know, it's science. It's where we're going. We have an aging population. We have greater consumer demand for nutrigenomics testing and personalized nutrition. It saves time. We have that quantified self-monitoring that people like. They want to take control of their own data and their own health. And we also have on our side is a rapid development in technology that will allow us to provide this to everyone indiscriminate of income, which is something that now you can, it's really the person that's health conscious that has disposable income that can actually do it. So 2017 was the year that consumer DNA testing blew up. So as you can see, there is also consumer demand for it. If you look at all the genetic testing companies, the number of people that have been tested, it's doubled in the last 2 years. So it's definitely where we're moving towards in terms of personalization. Now, because it's a big money business, there is consumer demand. There is a proliferation of companies that have popped up online all over the world offering genetic testing. So I just want to say a couple of things here is that it's really a largely unregulated market. There's a lot of companies making unsupported claims that are not using evidence-based science. And it's really important as healthcare professionals that you do the work and do the investigation to see who's behind it and actually look at the science. You know, most of us are trained to be able to look at the science. I'm always happy to be contacted by anyone wanting a bit of help interpreting and understanding it. But it's very important that you do the research and make sure that it's evidence-based and that you question what happens to your data. So a lot of people put in their name, their address, their email, send their sample in, and it's completely not de-identified. So you want to make sure, like, who has access to your data, who's looking at it, what are they doing with it, what's secure and what's not, because it is quite important that you have control of what happens to your genetic information. So for those of you that want to learn a little bit more about how to evaluate the evidence and determine if there is enough to make specific dietary advice for one particular genotype or other, I suggest you look at this review. It was done by the who's who in Nutrigenomics. If you look at the names here, you've got Ben Van Omen, Jose Ordovas, who's kind of the founder of the field of nutritional genomics, John Mathers, Mike Gibney, and you have our local Michael Fenech from Australia, Lynn Ferguson from New Zealand. It's just really a celebrity panel for me of authors that know what they're talking about. So moving forward, where are we going from here? Well, we know that there is robust evidence, at least for some markers, to make clear recommendations. Unlike epidemiological evidence in nutrition, often in nutrigenomics, when someone replicates a study, they get the exact same effect over and over and over. It's independent of ethnicity. So, for example, the prevalence of the actual genetic variation might be higher in one ethnicity, but what that gene does when it interacts with diet is going to be the same, irrespective of where you come from. We get that improved compliance and behavior change, where we have RCT evidence for. All the recommendations are actionable. It can also help improve epidemiological research. So, for example, it might help address some of the recall bias. It might help improve measures of exposure. It minimizes residual confounding, which you've seen in that caffeine study that I presented. Imagine if we ignore genetics. You can really see that clear effect of the confounding. You can help identify bioactives, but also mechanisms of action. So what's next? Well, basically, I predict that those guidelines are going to be completely outdated. At the moment, we're basically just fine-tuning. We all know we have to eat more vegetables, but the question is, how do you get people to eat more vegetables? How do you get them to eat better? And perhaps this is another tool in our toolkit to help people live healthier lifestyles. We know that more tests will be discovered and they'll be added to the panel of how we interpret results of genetics relating to nutrition. And I would predict that it will become routine testing, like it is for PKU, like it is for cholesterol. It would just be something that you're born knowing. For those of you who want to do a little bit of further education, to date, I've only ever found 2 evidence-based courses from the whole world, looking at nutrigenomics and nutritional genomics. And one's from Dietitians Canada, and you can click there and find the course on their continuing education. And there's also one from Monash University here in Australia that's really good as well, that gives you that overview introduction in nutritional genomics with a little bit more on the ethics and the regulation of it. So thank you very much for your time, and I'll hand over to Carlotta now, who's going to be talking a lot more about the science and the intricacies of the details on the med diet and extra virgin olive oil.
Thank you so much, Flavia, for that. I'm absolutely blown away by your knowledge in that area. That was so interesting. So thank you so much for that. Hopefully the sound is a bit better. My colleague just informed me that I was blowing everyone's ears off when I had my other headphones on, so I apologise for that. Hopefully the sound is better now. I would now like to introduce our second presenter, Dr. Carlotta Petti. She is a nutritionist and medical scientist. She is a qualified biologist nutritionist in Italy, is registered with the Nutrition Society of Australia and an active member of the Australian College of Nutritional and Environmental Medicine. After completing her master's degree in molecular genetics and PhD in cancer genetics in Europe, she obtained 2 additional postgraduate degrees in public health and human nutrition from Curtin and Deakin University. So bringing together her background in genetics, public health and nutrition, her focus is in the field of Nutrigenomics and Personalised Nutrition. She is currently the Scientific Director of Nutrigenomics at myDNA, where she leads the innovation of product development and directs research and education projects. So welcome, Carlotta, and I will now pass over to you.
Thank you, Abby. Thank you for the presentation, and thank you for inviting me to present today. And also thank you, Flavia, for an amazing presentation, always very interesting and insightful. So today I am particularly excited to be speaking here with you because I'll be talking about 2 of my favorite topics. So one being nutrigenomics. As Abby was saying, I've been passionate about genetics and nutrition and the connection with health since I started uni, so quite a few years ago, unfortunately. And now I'm fortunate enough to be involved in this new area of science, which is nutrigenomics. But also, as I was born in Italy, in particularly in Tuscany, I also have a very strong connection with extra virgin olive oil because it's really an integral part of my culture and, excuse the pun, of my DNA. So in Tuscany, you either grow your own olive trees and produce your own oil, or you always have a relative or a friend who can produce it for you. So it's quite amazing, and we can use it for cooking, for baking, for eating. We even use it on our skin. So you can understand my deep connection with this topic. Now, today, I would like to give you an overview of the research that links extra virgin olive oil with nutrigenomics. Now, it's not going to be a in-depth scientific description, but really the goal is to give a general overview of the science, which is quite large, and I'll start by touching on some interesting facts on fats and genes, and then I'll dive into the main character of the presentation, which is our extra virgin olive oil. And I'll present two different types of studies, the strictly speaking nutrigenomics and the nutrigenetics studies. So before I go into the detail, I'd like to spend a couple of words on this topic. Now, Flavia has already beautifully explained this, but I feel it's important to reinstate this concept so that I can help you understand the difference between the type of studies that I'm going to present. So nutritional genomics studies the interactions between food and genes, widely speaking. Often, nutritional genomics is also referred to as nutrigenomics. However, nutritional genomics is composed by two different areas of study, which is the strictly speaking nutrigenomics and the nutrigenetics. So you can understand how sometimes we can get a bit confused by talking about nutrigenomics, because we can refer to the overarching field of nutritional genomics as well as to one particular field of this area. So today, whenever I say nutrigenomics, I'm going to talk particularly about this category, which basically is looking specifically at the influence of nutrients on gene expression. And the type of studies that are mainly found in the nutrigenomics category involve things like epigenetics and all those omics technologies like transcriptomics, proteomics, and so on. Now, the ultimate goal for nutrigenomics is to identify the molecular fingerprint of health or disease. Instead, nutrigenetics looks at the other direction of this interaction, so how genetic variants influence the body response to different nutrients and what are the effects on health. The main type of studies that we use in nutrigenetics are generally genome-wide association studies or observational studies, but also dietary intervention, randomized control studies. And the goal is to identify the genetic susceptibility to a certain health outcome. Now, one question that genetics can help answering is, why do we eat fat? So there are some studies done on heritability of fat intake, and they show that the reason why we eat fatty foods is about 36 to 61% due to our genes. So although this is quite a big variability, which is mainly due to intrinsic limitation of some of the nutrition studies, as we know well, but it's an interesting fact to remember. So there's also quite a few genes that are related, or they have been suggested to be related to this mechanism, so they can mediate the genetic influence. And a couple of examples I've reported here in this slide, but I'm not going to go into such details today. Now, the next question is why we like fats or we don't. Until recently, we only knew about the existence of 5 tastes, which is sweet, sour, salty, bitter, and umami. But recently, there has been the discovery of a new taste, which has been called pinguis or oleogustus, that literally means the taste for fat. So our body can actually perceive fat as a distinct taste. Now, in this mechanism, there is a genetic component. And although the details are not fully elucidated yet, there is one gene candidate that has been recently reported as implicated in the mechanism of perceiving the fat taste. Now, this gene is CD36, and it was shown that fatty acids that are components of fat, fatty foods contain fatty acids and triglycerides. Now, the fatty acid components are able to bind to this receptor, CD36, which is expressed in the tongue and also in the nose. And this interaction is able to mediate the sensitivity for the fat taste. Now, interestingly, it's been found that obese women that carry a certain genetic variations in the CD36 gene can taste oleic acid 3 times less than non-carriers, which means they're also more prone to consume more olive oil. Now, I suspect I may have that variation. Now, fat intake can also modulate body size depending on specific variations. For example, some people may lose more weight with a high-fat diet or on a low-fat diet, and that's depending on their genetic variation for certain genes, like it's the case of the TFAP2 genetic variation, where GG lose more weight on a high-fat diet, but the AA genotype lose more weight on a low-fat diet. And also, in some cases, it has been shown that fat intake can modify the effect that the genotype would predict. Now, I would like to give an example of the FTO gene. The FTO gene is probably the most studied gene in the space of obesity, and there are a few SNPs that have been thoroughly studied and that have been associated with the risk of obesity. This is one of them. Now, what's interesting is that the FTO carriers of the risk allele for this SNP have a higher risk of obesity if their diet is high in fat, and in particular, if it's high in saturated fat. And in a similar fashion, there's also another SNP in the PPARG gene that basically the effect on BMI from a high-fat diet is only seen in the SNP carriers and when they have type 2 diabetes. Things are a bit different if they don't have type 2 diabetes, but yeah, I'm not going to go into these details, but this is, all a very useful information that shows how different people have different effects on their body mass in response to fat intake, which is based on genetic variants. Another interesting fact is that fat can directly control gene expression. So, in fact, fatty acids can physically bind to molecules that are nuclear receptors, and they're called PPRs, so peroxisome proliferator activated receptors. And these molecules act as transcription factors, and thus they control the transcription of the other genes downstream and activate certain pathways in the cell. To give you an example of this, I chose this Nutrigenomics study that looks at the effect of different types of fatty acids on gene expression. This study compares, in particular, 2 diets, one which is high in saturated fat, and it's called a butter-rich diet, and it compares that with a diet that is high in monounsaturated fat, and specifically it's high in olive oil. Now, they compare the gene expression in the adipose tissue, and what they find is that there is quite a bit of a difference in the gene expression profiles from the 2 diets. In particular, when you have a high saturated fat diet, you have a higher number of genes that are modulated in comparison to the monounsaturated fat, but also is the type of genes that are expressed that it's different. So the high saturated fat diet is actually modulating mainly pro-inflammatory genes, whereas the unsaturated fatty acid diet is modulating anti-inflammatory genes. Now, this can also provide an explanation, a molecular explanation of the inflammatory environment that commonly is seen and linked to obesity. So saturated fatty acids are pull ligands for PPRs. So when you have a diet that is rich in saturated fatty acids, the PPRs are downregulated, so they're not very much activated, and this can increase inflammation. So I think it's a good time now to introduce our special character of the presentation, which is the extra virgin olive oil. Now, there are a couple of things that are important to remember when we talk about EVO. First of all, there are different grades of olive oil, as you may be aware. So in the studies that I'm going to present, sometimes you hear about extra virgin olive oil or virgin olive oil or simply olive oil, and each of them will have different properties. If you're in Italy, you just need to say oil, and everyone knows that you talk about extra virgin olive oil because there's nothing else. But yeah, in these scientific studies, you have to understand a bit the difference. And the other thing is that to consider is that extra virgin olive oil has different components. So we know that it has a fatty acid component, and the main fatty acid is the oleic acid, but there are also other types of fatty acids in there. And the other big component is the bioactive molecules, which I'm gonna talk a little bit more later on. And lastly, the third thing to consider when we look at these studies is that often studies can talk interchangeably about Mediterranean diet or extra virgin olive oil, and that's simply because olive oil is really the hallmark of a Mediterranean diet. So you know that if you're having a Mediterranean diet, most likely you're going to have a high intake of olive oil. So there are many nutrigenomic studies that use these omics technologies that are very useful to clarify the molecular mechanisms of how extra virgin olive oil or Mediterranean diet produce the well-known health outcomes, which are usually a positive regulation of lipid metabolism, or action on immune inflammatory pathways, and also antioxidant activity. Now, one good example of transcriptomic studies is this study, which is based on the PREDIMED trial, and basically shows that the Mediterranean diet combined with high EVO diet specifically can modulate those pathways that are related to atherosclerosis and hypertension. There are many other studies that look this type of studies, so transcriptomic studies, very similar to this one. That and this is a bit of a summary from a systematic review of all these similar studies that show that there are quite a few distinct gene targets that have been identified as modulated by specifically EVO or Mediterranean diet or other components of extra virgin olive oil. So you can see here a bit of a summary of those genes. One of the most frequently modulated is TNF and interferon, and lots of genes that have to do with inflammation or oxidative stress or chronic disease. Now, in addition to studies that look at the effects of extra virgin olive oil and specifically the fatty acid components, there are also quite a few studies that look at the effects of the bioactive components of extra virgin olive oil. So here I just put some examples of the main components of the extra virgin olive oil. And in particular, I would like to mention one, which is one group, which is the biophenols one, because there is one component in particular, which is the hydroxytyrosol, HT, that has quite a few studies. So I thought it would be interesting to just spend a couple of words on that. Basically, lots of nutrigenomic studies done on hydroxytyrosol to see the effects on gene expression show that HT can modulate quite a few genes that are related to inflammatory pathways, so NF-κB, but also PPRG and TNF-alpha. But even more interestingly, it's been seen that HT can actually modulate a series of genes that are related to the NRF2 transcription factor pathway. So all this part of the research has suggested a mechanism for extra virgin olive oil and its antioxidant effects. So basically, the theory is that, at least in vivo, when you have low doses of biophenols, that can trigger protection against oxidative stress, not by having a direct effect as free radical scavenger, but basically these molecules, the bioactive components of the extra virgin olive oil, can indirectly have an antioxidant effect by targeting the NRF2 pathway. So that's an interesting explanation of how extra virgin olive oil can have these antioxidant effects. Now, so far we've seen mainly transcriptomic studies, but here's a bit of an overview of other omics studies. So there is some research on epigenetics, on proteomics, on metabolics, and these studies show effects of extra virgin olive oil on specific targets. And there is also a very new area in here, which is called myRNomics, apparently, that looks at microRNAs. So microRNAs are small sequences of RNA that can target genes and modulate their expression. Now, interestingly, it's been found that extra virgin olive oil can downregulate a specific microRNA, which is called mRNA 30AM, which is believed to be the marker for aging in the brain. So extra virgin olive oil could help slow down cognitive decline through this mechanism, which is, again, quite interesting finding. Now, following from these, there are also other studies that are starting to look at exogenous microRNAs as well. So olives coming from the olive tree will contain also microRNAs, like humans have produced microRNAs. So this new area of science is now trying to see how exogenous microRNAs coming from the olives that you can consume through your extra virgin olive oil, how these microRNAs are actually affecting human gene expression. So very fascinating, and I'm sure we will have more to read in the next few years. So in summary, nutrigenomic studies that mainly are in vitro, can be in vivo, or even some human studies, have started to identify potential mechanisms that are targets for components of extra virgin olive oil, and that may be responsible for the positive health effects of extra virgin olive oil, like anti-inflammatory, antioxidant, and cardiovascular protection. Now, we can now start talking a bit more about the nutrigenetic studies. Flavia has already introduced and explained a couple of studies there, very interesting. So I'm gonna just give you an overview again of some of these studies, but in particular, I'll start with this one because I think it's just a very simple example that can show how typically a good nutrigenetic study is designed. And then many other studies will follow the very similar structure. So the goal of this study was to investigate the interplay between interleukin-6, a SNP in the interleukin-6 gene, and the Mediterranean diet on body weight change. Now, this study is, again, based on the PREDIMED intervention, and it shows that at baseline, people who have the CC genotype for that SNP have the highest BMI, body weight and BMI. Now, after 3 years of intervention, which is the Mediterranean plus olive oil diet, the CC genotype displayed the greatest reduction in body weight. So basically what this says is that the CC carriers can benefit more from the anti-inflammatory properties of extra virgin olive oil, so more than the other two genotypes. Now, this type of studies really can allow us to identify in the population the portion of people who can benefit more from a certain intervention. So it's very powerful and useful information. And similar to this study, there are many others that I just put here as a simple summary that can look at different effects. So we saw now that study was about weight change. There are others that investigate the effects on weight change depending on different genes and different SNPs. There are also other studies that look at fat loss more specifically, or also studies that look at metabolic health and lipid profile, again involving different genes and different SNPs. Or the effects can be hypertension and cardiovascular disease in general. but also telomere length. So I just spent 2 words on that because it's something a little bit different. So this one study is, again, it's based on Predimed and shows that carriers of a risk allele in PPRG gene who follow a Mediterranean diet basically have lower telomere shortening. Now, as part of the normal aging, as you age, your telomeres are shortening. So basically, from this study, if we really want to put it in a very lay terms, we can say that if you have that specific genetic variation for PPRG and you eat a Mediterranean diet, you may even live longer, which is quite amazing. Now, in conclusion, we've seen some examples of nutrigenomic studies that are more explorative in nature and explain some of the mechanisms behind the effects of extra virgin olive oil and health. We've also seen some nutrigenetic studies that have shown the effects of a Evo-rich or a Mediterranean diet on certain health outcomes, depending on genetic variations. Now, as for everything, there is still more to be researched and proved, of course, but the information available so far is certainly interesting and provides a good understanding of the connections between extra virgin olive oil and also more in general fat and our genes and their effects on health. And this is just a summary of what I've presented today in line with our learning objectives. So I'm done. I thank you very much for your attention and I hope you enjoyed it. And I'm very happy to answer questions if we have some time. Thank you.
Thank you, Carlotta. That was fantastic. Such a wide range of knowledge there. I feel like this is definitely one I'm going to have to go back and watch, and thank God we have recorded it because, yeah, there's just so much information there. We do have a couple of questions that have come through. The first one is, are there any studies on effects of extra virgin olive oil on APOE genotypes?
Good question. No, I haven't come across specifically on that one, but it would be very interesting considering that obviously extra virgin olive oil is thought to have, you know, a good influence on cognitive decline or protecting against cognitive decline and having a neuroprotection role. So it would be very interesting. So I'm not aware of studies on that, but there could be, and I just haven't seen them. Flavia, have you seen any study on APOE?
Carlotta, yes, there are a few, but I don't know them off the top of my head. But I know that there have been some published looking interaction, I think, with atherosclerosis development.
Yeah, it would make sense because, yeah, it's definitely an interesting connection. So, yes, we'll look into that.
Thank you. The next question is, do you guys have, so what are the practical implications of all this information for dietitians who are in practice?
Sorry, can you repeat that again? So what are the implications for dietitians in practice?
Yeah.
So basically it's that I think it's really important for dietitians to be across this and to be able to offer nutrigenomics to some extent because it's the future of where nutrition is going. We're all trained in genetics. It's part of what we are trained in and are capable of doing. You can do courses online, like from Dietitians Canada, from Monash Uni, to further your CPD on that topic, to build the confidence. But like anything else, like, I mean, I've done about 300 consults using genetic testing in practice. And it's just like when you're a new grad dietitian, you know, it gets better and better and better the more experience that you have, because you can then pinpoint things quite easily. You're more across the science. But it's something that I think we have to embrace it rather than be like, oh, too hard, too complicated. Let's not. We already use celiac disease in our scope of practice, lactose intolerance, PKU. I mean, there are things that are part of our curriculum that we're trained in. So it's just thinking about the curriculum being expanded to now include those extra things where the science has evolved. So in summary, yes, it's in our scope of practice. It's in the Dietitians Canada scope of practice, which is very similar to ours in Australia. So it's something that I really believe we need to do a bit more CPD and to be confident in offering and interpreting the science, because it's your consumers are going to request it. They're going to come to you going, I got this tested online, because it's available direct to consumer, and they're going to say, interpret it. And if you can't, then you're basically losing that clientele to some other healthcare professional. And as dietitians, we need to embrace that area, because we're the experts.
And I agree, as you were saying at the beginning, Flavia, that it's very important to, for everyone who wants to use this information in their private practice to really, be empowered, so get to know the science and understanding the science themselves, because that's the most powerful tool that then you can use to interpret any study and make your recommendations based on that. And
anecdotally too, like from my experience, 99% of my time is in research. It's not in private practice, but I do for some corporate clients and friends of friends and family. But it's what I love about it, and the reason why I kind of left private practice, is because I felt like, same person, same problem, same recommendation. I felt like I wasn't really personalizing to the extent that I wanted to see results. And from using genetic testing, you get hassled. When's my test ready? Can I come see you? Which we generally don't get, people chasing you for your consult. Two, it takes a little bit of the guesswork out from the trial and error if you're doing weight loss, or you can straight away go to a particular recommendation that takes genetics into account as well as lifestyle. And so you just have a bit more confidence as a nutrition professional to know that perhaps what you're recommending is going to work, but also knowing that that RCT evidence that exists on behavior change. So you've got that as well, that it's likely going to help them change their behavior, because, we all want to know more about ourselves. That's what we're really interested in. How can I better myself? And it's so specific to them that it helps give that motivation to come and get help and fine-tune the diet. But it is, a cost. At the moment, most genetic tests are between $100 to $300. So it's not cheap. It's the consumer that's, aware of their health and that has the income to pay for it. So at the moment, it's quite specific to that consumer profile.
Yeah. And as you said, it's probably, the future is the way where, not just the future, is the way where we are going, the way that it's already happening. And if you think about it, if you really think about the whole story of the one size fits all, that now we realize it doesn't, and this is giving the real one of the explanations as to why that didn't work, then as a practitioner, I guess you can understand how powerful is this information, which obviously it's not the only tool that you need to use, but it's definitely a nice, a very useful addition to your practice, because it makes your practice even more powerful. You can combine all the lifestyle factors and all the individualized information that you have about the customer with the genetics. So it's really the way to the real personalized nutrition and personalized advice.
And I mean, once it's hard to stick to the general dietary advice. So for example, Australia doesn't have kind of limits on coffee consumption, but internationally it's about 3 to 4 cups. So if I were to tell someone no more than 4 cups, I'm basically potentially killing half of you and then benefiting the other half. So basically if you don't know if you're a fast with low caffeine metabolizer, the limit should really be 2. But public health recommendations are 3 to 4. So it's really important to take genetics into account, even from the research level.
I agree.
It's so weird not having feedback, right? Are you guys nodding? Or going no?
It'll be good to see.
Any other questions?
Is there any other questions? Abby, you're still there? Maybe we lost her.
I am here, so can you guys hear me?
Yes.
Oh, perfect. I must have lost you for a little bit there. So thank you. That kind of does bring us to the end. There were a couple more questions, but I'll make sure I send them to both Carlotta and Flavia and get them answered for everyone who was here today, and we can send them in the post email. I just wanted to quickly mention some exciting news from the Olive Wellness Institute. We are now launching the Olive Wellness podcast. The launch date is 3rd of September, and we'll be having a new podcast available every 2 weeks. And our first guest is Dr. Joanna McMillan. So please, if you could subscribe to that, some fabulous information coming through there. And yeah, thank you so much again, Flavia and Carlotta, for your time and wealth of knowledge and information.
Thank you. Thanks for having me.
It was a pleasure.
Thank you. I'll quickly pop up the survey now, and it'll stay up for a few minutes. So if anyone who's still online could please complete that. And thank you.